Good morning, everyone. Welcome to today's MPG primer, and also welcome back to MPG after the lovely and all still cold and snowy winter break. So Dr. Hilary Finucane is a distinguished statistical geneticist renowned for her innovative work in understanding the genetic basis of complex traits and diseases. Hilary is an assistant professor at Harvard Medical School and Massachusetts General Hospital, as well as an associate member of the Broad Institute. Hilary earned her PhD in applied mathematics at MIT. Did I say MIT? I don't know what I said before, where she was supported by a prestigious Hertz Foundation Fellowship. Hilary also holds a Master's of Science in Theoretical Computer Science from the Weizmann Institute of Science and a Bachelor of Arts in Mathematics from Harvard. Among her many honors, she is a recipient of the National Institutes of Health Directors Early Independence Award. The Finucane Lab develops and applies novel statistical and computational methods to integrate genetic, molecular, and functional data to elucidate how genetic variation contributes to disease risk and thereby advance our understanding of human health and disease. Hilary has said that you can take questions. She'll take questions throughout the talk. And also what's really interesting about her talk today, it's going to be a chalk talk. So this is something we're kind of demoing out with Hilary. And if please give us your feedback after the talk or later in the year about what you thought of this different way to engage with the material. All right, take it away, Hilary.
Thanks so much, Nicole. All right. Hi, everyone. I'm looking forward to trying out this talk format. And could we do the screen? Awesome. Okay, great. My plan today is not to go deep on any one topic. I think most everything that I'm going to touch on, there's a separate primer that actually goes deep. But my goal instead is to kind of give an overview that shows how these different things are connected. And it is kind of like a framework that, in my experience, has really helped clarify some things that feel like they're really sticky technical stuff, when actually, if you kind of back up and write down the model, then things tend to make a lot more sense. And like Nicole said, please do ask questions, even if it's just like, oh, I didn't understand. Can you say that again? I think this group is small enough that you can just raise your hand and ask. Okay, so what are we going to talk about today? So today what we're going to talk about is what are uses of models of the form yi equals sum over j in S, xi j beta j plus epsilon I, where yi is the phenotype of an individual I, xij is the genotype of individual I at a variant j. Beta j is the effect size of variant j. S is a set of variance, and epsilon I is a residual. One thing I like about chalk talks, by the way, is they take the pace down a little bit, and then that gives everybody a chance to kind of think about what it is that they find confusing. So first, I'm going to make one note about this. And that's that this, crucially, this is kind of one of the main punchlines of today. This depends on the set S. So if we're going to write down a linear model, so to give kind of an overview of what I'm about to say, this kind of model shows up in a lot of places. So if S just has one SNP, and you're fitting us like one model separately for every single SNP, then beta j, this is no sampling noise, no error or anything. The true beta j that you're trying to estimate is the marginal effect size. And so, for example, if SNP j is an LD with some other SNP, and that other SNP is causal and SNP j is not causal, the true beta is still going to be non-zero. On the other hand, If you have a really dense set of SNPs that's got every possible variant in some locus, then you can, under some extra assumptions, start to interpret beta J as true causal effect sizes. Maybe you're doing polygenic risk prediction using a model that's something like this, how you choose to incorporate effect sizes is going to depend a lot on whether you've LD pruned, for example, or have not LD pruned. What this model is telling us, and specifically what the effect sizes in the model are, is going to entirely depend on what set we put there, because these are joint effect sizes. I'm also going to put a couple of assumptions down, or notes, I guess. So I'm only going to talk today about quantitative traits. So you can think of height as an example. A lot of the methods that work for quantitative traits really directly carry over to disease and other case control traits. I have come to appreciate that this is in some ways specific to this thing where we're working with common diseases and complex traits, because our effect sizes tend to be pretty small. And so a lot of, there have been some derivations of why it's okay to pretend like our binary traits are continuous. And usually there's a Taylor approximation that works really well somewhere in there. And Taylor approximations work really well when your effect sizes are really small. So that's kind of intuitively why I'm going to only talk about quantitative traits, but why basically every method I'm going to write down has been applied very successfully for case control traits. And then the other thing is, just for simplicity, I'm going to omit covariates. In practice, when this actually gets used, then there's a bunch of covariates here, age and sex and ancestry and so forth. But just for simplicity, I'm not going to write those down. Okay. So my plan is to talk about, there's 5 more panels on this board. I'm going to talk about 5 instantiations of this model to kind of emphasize the differences in how this exact same basic equation can be useful for all of these different things. Okay, so to start with, let's talk about basic association mapping, i.e. GWAS. Okay, so what's the goal here? As I said earlier, the goal is not to identify the causal variants. The goal is to identify regions of the genome that are associated to a phenotype. So let me write that down. Identify regions of the genome associated phenotype. And what's our particular instantiation of this model? Well, as I said before, there's no summation. We're just going to say xij beta j plus epsilon I. And there's going to be a different model for every SNP. And what are we interested in when we do this? So you can think of this as when we fit this model, what is the thing that we actually care about and that we want to be well calibrated? And for the most part, what we care about is a p-value for the null hypothesis that beta j is 0. And so the intuition there is there a causal variant in LD with variant J? We also are often interested in estimates of the marginal effect size of SNP J. So let me see if there's any other notes about that. Yeah, so I think that that's, in a GWAS, what we do is we go one SNP at a time, we fit this model, and then we look for p-values that are significant after multiple testing. One thing that I'll mention is that the set of results from a GWAS are often referred to as summary statistics. I'll come back to this later in the talk, but a lot of the methods that we're using for fine mapping or for heritability estimation of various sorts or for risk prediction can be done using summary statistics. That's right. Okay, so now let's talk about methods. And there's two general categories of methods here. In one case, we have, actually, I'm going to make this the vector epsilon. So So I haven't specifically talked about what the distribution of this residual is. And in the simplest case, we're just going to assume that it's IID normal for all individuals. So this is kind of this most standard linear regression that you could do. In this case, we'll just do ordinary least squares. And then there's another family of methods in which you make a different assumption. Now you have two components. So if we want to instead assume that our residuals can be correlated across individuals and that those correlations are reflected in a relatedness matrix, then now we're talking about linear mixed models. And there's been a whole lot of work on linear mixed models and how to, first of all, estimate these parameters, then how to estimate the parameters in a way that increases your power, and then how to estimate them in a computationally efficient way. So this is kind of an interesting subfield because the problem gets so much harder once you make it complicated in this way. So that's all I'm going to say about GWAS for these purposes. And maybe I'll pause before I move on to the next section and ask if there's any questions so far. Yes. Yeah, so there's been a lot of work on that. One common thing to do is inverse rank transform it, where I guess there's a couple of ways to think about this. One way to think about it is that there's some true underlying heritable trait that's normally distributed, and that got monotonically transformed to give you some weird distribution. And so then you can recover this original maximally heritable trait by just using the ranks and forcing it to be normally distributed. And I think that has been surprisingly successful in a lot of cases, even though it's like a pretty strong assumption. But a lot of folks, will look at the data, take logs, remove outliers, the kind of stuff that I think is pretty standard in linear models. There has been some work. So several years back, there was some work by Niccolo Fusi at Microsoft Research learning the underlying monotonic transformation that maximized the heritability, which I thought was kind of an interesting way to think about it. So yeah, but I think that this intuition that, well, okay, and also I guess if we're going to move beyond quantitative to, case control or count or ordinal or that kind of thing, then there's, a large family of methods for dealing with that kind of thing as well. Yeah. Awesome. All right. So next I'm going to talk about, let me give this a box so that it stands out. So next I'm going to talk about fine mapping. And what is our goal when we're fine mapping? Now we actually want to identify the causal variance. To go back to this for a second, this is kind of genius, right? Like it's saying we're going to start with a problem that's an easier problem to solve that can be solved with a simpler statistical method. And then it's really robust. When we're trying to estimate these marginal association statistics, the result for one SNP, it doesn't depend on the other SNPs. And empirically, it's been true that we've been able to replicate results from one study in another study quite reliably. So this now is like this rock solid, it's more or less a solved problem, how we do basic association mapping. And the reason that it's so thoroughly established and so reliable is because we're not trying to identify the causal variance yet. We've set for ourselves a goal that can be solved by fitting one SNP at a time, and we've gotten really good at that, as a field, I mean. But But now we want to identify the causal variants. This is the more interesting goal, and it is way, way, way harder. Okay, so why is it way harder? What's the model that we're fitting? The model that we're fitting now so this looks very similar, but now what we have is so dense is the key word here. Right. At a locus. So now what we're going to do is we're going to specify one locus. We're not going to fit one model for the entire genome because we don't need to do that here. Instead, we're going to choose a locus at which there's an association. We think that there is a causal variant somewhere here. We want to know which one is the causal variant. So we're going to fit a model just at that locus. How you define loci, that can be a bit of a thing. I won't go into that. And now what we're going to do is we're going to have as dense as possible a set of variants at that locus, because in order to have the interpretation that we want, which is that beta J is the true causal effect size, there's a number of assumptions that have to be satisfied. And one of them is the true causal variant has to be one of the ones that we included in our model. And so let's see. So okay, so what are we interested in? Usually, we're now going to think about this in a Bayesian framework, because the kind of an intuitive thing to think about is that we want to know what the probability is that beta J is not 0 given all of our data. So we want to know what's the probability that that the true underlying causal effect is non-zero, i.e., that this variant is a causal variant. And so why is this difficult? One reason this is difficult is because of LD. If you have two SNPs that are in perfect LD, then there's no way to have total confidence that one of them is causal and not the other one. You just can't. It is the same information encoded twice. You can't disentangle that. It's nice in that case to be Bayesian, because then you can say, if I think only one of them is causal, if your prior encodes some kind of sparsity, then your posterior probabilities will be close to 50-50 for the two variants that are in perfect LD. There have been other folks who have thought about this from a frequentist point of view, thinking about sets of SNPs that are likely to contain a causal SNP, which is also a useful framework. But a lot of the methods that are most commonly used right now are Bayesian. So let's talk about methods. And this, again, I think, is the topic of an entire primer. So I won't spend too much time on it. But I will say that there's several families of methods, and I'll write down some classifications. So one is single causal variant methods. So this is the exception to what I was just saying. If you're willing to assume that there's only one causal variant in the locus, then everything gets really easy. And I won't do the derivation on the board, but you can write down, you're now looking for a posterior over size of S configurations. So you're just there's S possible outcomes because you know exactly one of these variants is causal because that's what you assumed. And now you just need to know what the posterior probability is of each of those outcomes. So things are super easy in that case. The problem is once you get into multiple causal variants. And of course, the tricky part is we know that there are multiple causal variants at a very large number of our loci. So it's very nice and convenient that we can do one causal variant fine mapping, but that loses a lot because we know that the assumption is violated in a lot of cases. So now let's try to make some more realistic assumptions. Let's say that we want to have multiple causal variants. So this is where things get really tricky, right? If you wanted to say, I'm not going to put any restriction on the number of causal variants, I'm going to put some kind of prior on the probability that, that a variant is causal, and then I'm going to do exact Bayesian inference, then the number of states in your prior now is going to be exponential in the size of S, because there's any possible subset of S could be your set of causal variants, and now you need to get posteriors for every possible subset of S. So exact Bayesian inference is very challenging computationally. So I'm going to say exact Bayesian inference is hard. You know, folks who do this kind of thing really hate discrete spaces, and this is very as discrete as they come. Okay, so what are some solutions that people have done? One thing that folks have done, so this was mostly in the early days, is just to restrict the problem, to restrict both the size of S and also restrict the number of causal variants. So we're not going to say one causal variant, we're going to say 3 causal variants, which already actually that helps a lot. But the reason that this can be tricky is because if you're still going to do, you know, exact enumeration of all possible configurations, then you need to make S really small. The reason that that's tough is because if you, in your model, you leave out a causal variant that has LD to the variants that are in your model, then that's a really bad model violation. So what are some other things that people have done? One thing that people have done is really, I'll say, like, explore a subset of the posterior space. There's been, so now we get into like really smart and creative things that a lot of people have done in previous years in a lot of different groups. So one thing has been to do deterministic search algorithms to try to find the configurations of causal variants that cover most of the posterior probability but are of a tractable size. There's also been stochastic versions of that using randomness in order to design an algorithm that also is exploring the sets of the, you know, the large subsets of the posterior probability. And so we're not trying now to accurately write down the posterior probability of every very unlikely configuration. We only care about the most likely ones, and we have some kind of handle on how we might go about finding the most likely configurations. Then another thing that we might want to do is conditional analysis. So I guess I should have put this above, because this is also before we get into large-scale approximate Bayesian algorithms. Conditional analysis can be, we assume that the most significant SNP reflects one causal variant, and that once we condition on that, then we can fine map the next one. And there's other ways that folks have tried to split into signals. So maybe we have a locus, maybe we think the locus has multiple causal variants, but we think there's literally enough LD among the different causal variants that we're going to be able to take the locus, split it up into, you know, in some way that's informed by the data, split it up into different signals and apply our fine mapping to each of the different signals. And then the last one that I'll write is iterative conditional analysis, which turns out to be conditional analysis, which turns out to be equivalent to variational inference. Where your variational assumption now is independence of the locations of each of the causal SNPs rather than independence of each of the beta Js. And so that's kind of some of the different ways that different folks have solved this problem, where you want to get a Bayesian posterior, you want the Bayesian posterior to allow for the possibility of multiple causal variants, but you don't want to enumerate 2 to the S possible configurations. And I'm not writing down specific references here. I think this one was Caviar. This is DAPG, or also FineMap. This is conditional analysis, and this one is Suzy. And the exact references are in notes that I'll post online if anybody's interested. And so then what are some other approaches that folks have taken to this, kind of in combination with 1 or 2? One is to incorporate functional data, functional data. So here, maybe what we want to do is say that, well, we know that coding variants are more likely to be causal than non-coding variants, or we know that conserved variants are more likely to be causal than unconserved variants. And so it would be stupid to just throw that information away. So we've got to incorporate it in the form of a prior so that we wind up getting posteriors that reflect that, that fact. And what's interesting there is, well, exactly how much should you upweight coding variants compared to non-coding variants? You could kind of subjectively say, well, my prior is, 5 or 10 or whatever. Or there have been a couple of different algorithms for using the data either in the locus itself or across many loci to inform what the relative weights should be of these variants that have these different properties. And then the last thing that I'll write is just that cross-population analysis can be complicated here. And that's because, well, for a couple of different reasons. One is a lot of the analyses are most easily done by splitting apart the summary statistics from the LD, and the LD can be different in different populations, and so you have to be a little bit thoughtful when you do that kind of analysis. And there's been some methods development work in a couple of different places trying to do that in as rigorous as possible a way. And it seems like it really does give a big boost for power when that can be done successfully.
Can I ask a question, Hilary?
Yeah, please.
What do you think is more powerful, incorporating more diversity of people in your GWAS or just increasing the N of your homogeneous population?
Yeah, I think that's a great question. I think that in theory, if we had perfect methods, then diversity, well, okay, I guess it's a trade-off, right? Like just, or, it depends a lot on where you are. And actually, a paper from Hailiang's group, I think if I'm remembering right, it has some figures about exactly that question, like at what point does it become a better idea to switch from just increasing your sample size to increasing your diversity? Because if you've got a really small sample size and if it's too diverse ancestrally to be able to do a good job of accounting for ancestry and potential confounding by ancestry, then you're really in trouble, right? So I think that, yeah, and we've seen examples before of data sets where because they're studying some molecular phenotype, the sample sizes are much smaller. But then if you don't yet have the methods to account for the confounding that you might see, then that can create problems. On the other hand, if you can choose between two big GWAS of the same ancestry or two GWAS of different ancestries, then the fact that you've got this different LD really helps break down, this main challenge of fine mapping, which is that LD makes it difficult. And so I think that we need the statistical methods so that the answer is always diversity, and I don't think our statistical methods are quite there yet. So I don't think that's always the answer. Yeah. Yeah. Yeah.
Yeah, thanks. But originally, how do you define it? Is like you have a GWAS signal and you say, okay, I just take, I don't know, 100 kb on both sides, or do you use LD, or like what is the, I guess the easiest is by size, but I feel it's not the smartest way. So I was wondering what the state of the art way to do it would be.
Yeah, that's a good question. So I think different folks have done this differently. One way that you can do it is, so one way you can do it is by LD. That is computationally intensive. One argument for doing it by size is if you do it by size and you do it big enough, then if you've got your locus that you're fine mapping right here, and now you include everything that could plausibly be an LD with it, then maybe that'll be tricky, if you're trying to interpret really strong signals out at the edges of your locus. But really what you care about is what's in the middle. And so you'll get much more accurate results for the middle as long as your locus includes the things that are in LD with the locus that you most care about. But I think I'm trying to think if I can think of any references off the top of my head for, but I think people have done it in both ways. Yeah. Yeah.
On that point, it will be a trade-off, right? Because at some point, if you include too many SNPs, you might be violating the single causal variant hypothesis, right?
Yeah, so definitely. Yeah, single causal variant fine mapping on really large loci, I agree, would be fraught.
And the reason why I'm just running around is just for the people online. It's a little bit, it can be hard to hear. So there's mics up here, or I can get more exercise this morning.
Great. So one other thing that I'll say that my group has done some work on is once you start to, if you say, okay, I've got my model, I've got the data, I've got my inference algorithm, and now I'm going to get a posterior, that's one thing. If you say, I have some intuition for what I want to be able to do, but now what I actually have access to is summary statistics from a meta-analysis, then the number of, we're always making assumptions, we're always violating the assumptions. It turns out that the assumptions that you're violating when you find map from summary statistics from a meta-analysis can sometimes be so bad that you wind up getting really unreliable results. So when you're doing Bayesian inference, a lot of times the initial derivation will be, I'm gonna say I have all of my y's, I have all of my x's, and now I'm gonna get a posterior conditioned on y and x. And then you can do derivations showing that, well, actually everything only depends on x transpose x and x transpose y, which are closely related to summary statistics in LD. And so now my math says I can use summary statistics in LD. And then you make a huge leap to say, I don't actually have the exact LD matrix from my specific set of genotypes. I have a reference panel. And when I talk about heritability, I'll say why that sometimes works in the case of heritability. For fine mapping, many different works, including works from our group, but also from other folks, have shown that that can really cause issues with fine mapping. And when you're doing a meta-analysis, then on top of that, often you have several different LD matrices from several different cohorts, none of which you have access to. And then once you combine that with other types of heterogeneity where you've got different genotyping in different cohorts and different imputation panels, then the set of complications that you're brushing under the rug becomes problematically large, even though there's always complications that we have to brush under the rug to do inference under any model. So that's one thing that I'll say is doing this from summary statistics can be difficult if the summary statistics are not exactly derived from your cohort. Okay, so now I'll go over here. Oh, and I will quickly talk about heritability. Okay. Sorry, that was my bad. The clock's been there the whole time. Okay, so I'm going to talk about heritability based analyses. So this covers all three of these. So there's basic heritability estimation, heritability estimation, genetic correlation, and partitioned heritability. And the reason that I'm writing them all under the same category is because they use the same S, basically. And so in particular here, what we've got is, again, sum over J and S, Xi, J, beta J, epsilon I, and this time S is a set, and I'll call this heritability. G. And what we're interested in, so this is the model. What we're interested in is what is the variance of, I'll call it G. Where so if we say that this is a genetic component due to genotyped SNPs plus some residual that has all the rest of the genetics that's not tagged by genotyped SNPs, like ultra rare variants or things like that, plus all the environmental components, then SNP heritability is asking how much of the variation in the phenotype, what proportion of that variation can be explained by genotyped SNPs. And one intuition for what this quantity is that we're trying to estimate is, if some oracle told me what the true betas were, then what's the best possible prediction R squared that I could get if all I have access to is a set of genotyped SNPs. So one thing that I'll notice, the betas here aren't actually that interesting. They're not marginal. They're also not causal. The set of SNPs is not particularly dense. And so this is good because it means that we can now start to do genome-wide modeling. And genome-wide modeling is good because now we can estimate this quantity that we care about, which is just the total amount of variation that's explained by the set of SNPs. It also allows us to do genome-wide estimation of genetic correlation, which is a closely related model, and also of partitioned heritability, where we want to know what's the contribution, for example, of SNPs in brain enhancers genome-wide. And now there's no particular locus that we care about so much that we want to throw all of our computational resources into, fitting some really dense set of SNPs. But we also don't want to just do marginal analysis. And so instead, there's kind of this intermediate thing where the betas themselves are not that interesting, but the model as a whole is still going to allow us to estimate these genome-wide quantities that are really interesting. And so with genetic correlation, maybe I won't write down, well, yeah, no, I think I have time. Okay, so with genetic correlation, what we want to know is, so the goal is how much do these two traits have shared genetic basis. And the model that we're going to write now is, I missed an I here. We now have two phenotypes. We have YIA and we have YIB. So this might be, I don't know, your height and your BMI or something like that. And now this is going to be and we have phenotype-specific effect sizes, of course, because that's what we got to have. And so if we call this, for example, GI B, then now we can say what we want to know is what's the correlation between the genetic component for phenotype A, the genetic component for phenotype B. And yeah, and so for partitioned heritability, the goal is explained by this set of SNPs. And so, for example, if this set of SNPs is all of the SNPs that lie within 100 kb of a gene that's highly expressed in a certain tissue, then this type of analysis, being able to estimate this type of heritability, can start to tell you about which tissues are most relevant for particular phenotypes from their genetic signal. And so here, the model that we're fitting is to now start to break up this genetic term into multiple genetic terms. So you can say YI would be and so if we call this and then what we're interested in is so out of all of the genetic signal, what proportion is explained by this set of SNPs, which, as I said, you'll choose to be some interesting set of SNPs where you care about how important they are for the phenotype. And so then to talk about methods, so now I'm going to talk about methods for all three, because the methods are shared. In general, when someone develops a method for one of them, then you can kind of apply the method to solve these different ones. And so the most of the methods assume that beta is random. So they add an extra layer to this model where they say beta is drawn from some distribution. And now we're going to relate what we are estimand properties of the distribution of beta, which I won't go into much more detail about, but I'll just say, once you say beta is random, then you can say either I am going to require full genotype and phenotype information, or just do summary statistics. And we can make like a table here, where now the rows of the table are going to be, I want to do something that's likelihood based, or I want to do something that's moments based, or I want to do something that's Bayesian. And it turns out, so we've got GCTA, we've got HE regression, and this is not comprehensive, of course, HE regression, and then this older method BSLMM. And over here, the last time I gave this talk, there wasn't a method in this slot, but now there is. LDSC and RSS. So there's lots of different methods for solving this kind of large family of problems. But for a lot of them, you can think of what kind of data are we requiring, and then are we gonna do something that's full likelihood based only on moments or Bayesian. And there can be advantages and disadvantages of each of those different approaches. And this all assumes that beta is random. So then separately, there was some work trying to actually solve it for when beta is fixed, which makes it a little trickier in a lot of ways. And so the main method that I know that goes under that category is the Hess method. So then if we want to come back and say, why does all of this matter for example, risk prediction? So a lot of times with risk prediction, what you want to do is write down a linear model that looks a lot like the model that we're writing down over and over and over again. And then you want to include some estimates of beta. And I think that, for example, if you're trying to decide whether to LD prune or not, well, if you got your estimates of beta from a model that's different for every SNP, then in order for that to make sense in the context of a larger model where you're summing across SNPs, then you need to show that it's okay to plug in, these marginal effects into what's effectively a joint model for risk prediction. That turns out to be okay only if there's no LD among the different SNPs. But a lot of, if you're interested in, for example, cross-population risk prediction, then now the question of whether you're interpreting your betas as true causal betas or marginal betas, or betas that are somewhere in between. They're not true causal betas. They include some effect from the tagged SNPs that are not included in the model. So marginal, anything that's not truly causal is going to be influenced by the LD patterns of the population. So if you're trying to do cross-population risk prediction, just as an example application, then you have to think really carefully about whether the true underlying betas, are they causal, in which case you can assume they're the same across populations pretty safely, depending on how you're scaling, or when you say beta, do you actually mean some combination of effects of variants that are tagged, in which case now the true betas are going to vary across population because LD and allele frequency vary across population. So sometimes it can just be helpful to think carefully about, I'm going to write down this linear model, and now I'm going to think carefully about even before I start estimating anything, I just need to think about what the beta itself means in that model. Now we can start estimating and worrying about power and noise and bias and so forth. But sometimes even just starting before you do all of that with saying, when I say beta, what do I mean, can be really helpful. So I'm going to stop there, and I am happy to take 2 minutes of questions.
For the genetic correlation, is the assumption that you have the full set of traits associated with each phenotype? Because if you see that there's one shared variant, but you don't know about 50 other different variants, wouldn't you think that they're perfectly correlated, these two traits?
Wait, so can you say that again? You're wondering if you do GWAS on 2 traits and you see there's one locus that's shared?
Well, maybe I'm misunderstanding the model, but if you have one shared variant between 2 traits, but that there's 50 different variants between the traits that you don't know about,
Right,
you
right,
might
right.
think that your traits are perfectly genetically correlated.
Oh, I see what you're saying.
Yeah.
So I think that in that case, you would get something that's not a perfect correlation because a lot of your betas would be 0 here. Right, so like you can imagine you've got like beta A and beta B. And maybe you've got something where you've got like a shared, maybe it has the same effect size for the 2 variants. But then you've got, okay, I'm just going to use 1s and 0s. You've got a bunch of 1s and 0s here, and then you've got a bunch of 1s and 0s here. So that this correlation is going to be really far from 1. And when you start like, when you plug this into this and you get this, then the correlation is going to be far from 1.
I mean, in the case when you don't know those other betas, you only have the one, the other.
Yeah. So this gets, again, at the difference between what we're trying to estimate and what we are estimating. So I think you'll agree what we're trying to estimate is definitely far from 0. The methods for doing this don't begin by looking only at genome-wide significant loci. So they actually do incorporate data genome-wide, whether or not the locus has reached genome-wide significance. And so then I guess I really haven't gone into any detail about this, but once you write down, let's say our model is that the ordered pair beta JA, beta JB is drawn IID from some bivariate normal distribution where the IID is over SNPs J. And now we want to infer parameters of that distribution. So you can start with that as your model and then derive properties of the distribution of X and Y that let you then do valid inference on the properties of beta to begin with.
I think with that, we're out of time, but I'm sure Hilary could take a few questions afterwards. So let's thank her one more time.